Hi, and welcome to the workshop, where we'll explore some new tools available for the food industry. Today, we'll specifically focus on an innovative approach called quantum analytics, and how this technology can be leveraged by the food sector to develop more specialized and personalized products. We'll start by looking at some background information to understand the foundations of this approach. Then, in the second part of the workshop, we'll show how this technology is practically applied, what it can do, and what kind of outputs it can generate. My name is Tanya Begar. I'm a microbiologist by background, and later I completed a PhD in biomedicine. Everything related to biology and the interaction between the environment and our biology is essentially my area of expertise, my professional playground, so to speak. Let's start with the very basics. What defines a living organism? A living being is typically characterized by three fundamental traits. It must have an active metabolism, it must be capable of growth, and it must be able to reproduce. These basic characteristics that define life become even more significant in highly evolved life forms, such as human beings. What sets us apart is our complex biological composition and the high degree of organization present at every level, from molecules to cells to tissues. Every biological molecule, structure, and organ has a specific role and function. Living organisms extract energy from their environment in different ways. Plants, for example, convert sunlight into sugars through photosynthesis, while we extract energy from the nutrients we consume and the oxygen we breathe. As mentioned earlier, one of the key characteristics of life is the ability to reproduce. In the first part of this lecture, we'll gradually dive deeper into understanding how our biology is structured and organized. If we now take a closer look at the molecular level, we see that the chemical structures of the molecules that make up all living organisms are remarkably similar. Basic chemical elements combine to form a wide variety of molecules, which in turn build cells, tissues, specific organs, organ systems, and ultimately, the entire human body. When we talk about the processes occurring within the cell and the body as a whole, it is essential to understand how these molecules are composed, what their functions are, and how bioenergetic processes operate both within individual cells and in communication between them. When examining the composition of life, we must always begin with the simplest components, the basic chemical elements, and progress toward increasingly complex structures. In biology, this hierarchical structure is very precisely defined. We start with fundamental chemical elements, which combine to form basic biomolecules. These biomolecules then assemble into essential cellular components, such as membranes, genomic material, and mitochondria. This stepwise organization ultimately gives rise to the structure of a single cell, which in turn serves as the building block for the entire organism. We begin at the level of the cell. Each cell is composed of various cellular compartments and specialized organelles. These structures are built from nucleic acids, proteins, saccharides, and lipids, which in turn are composed of basic chemical elements. The elements highlighted in red on this periodic table represent those most abundant in the human body. The yellow-marked elements are commonly referred to as minerals or microelements. Those marked in blue are required only in minute quantities and can become toxic at higher concentrations. However, current understanding shows that virtually the entire periodic table is relevant for human biology, with different elements needed in highly specific and diverse concentrations to support life processes. If we examine the elemental composition of the human body by mass, we find that oxygen is the most abundant element, followed by slightly smaller amounts of carbon, then hydrogen, and so on. For comparison, if we recall the elements marked in yellow on the periodic table, which represent essential minerals, we can observe that their total quantity in an adult human body typically amounts to only 100 or a few grams, or even less in some cases. As mentioned, the majority of the human body is composed of oxygen, followed by smaller amounts of carbon, hydrogen, and nitrogen. All other elements combined make up only a very small fraction of the body's total mass. Interestingly, the human body is primarily composed of elements that are also abundant in the Earth's crust. In essence, we are made up largely of the same elements that are prevalent in our natural environment. These 6 elements represent the fundamental matrix of life. They are predominant in biological systems because of their highly favorable structural properties and broad chemical versatility. For instance, carbon can form 4 covalent bonds, which allows for the creation of stable, low-entropy molecular structures. As we will see later, These 6 elements serve as the essential building blocks of all biological molecules. If we examine the structure of a basic atom, in the nucleus of the atom we have protons and neutrons, and we have electrons orbiting around. This fundamental atomic structure is consistent across all forms of life within the phylogenetic tree. Today, we understand that humans represent only a small fraction, illustrated here as the small green branch, of the modern phylogenetic tree. In fact, the vast majority of life on Earth belongs to the bacterial and fungal kingdoms. However, in this presentation, our focus is specifically on the interaction between the human body and food, that is, substances originating from our surrounding environment. When we take a closer look at the human body, we find that it is composed of approximately 35 billion cells. If we were able to observe these cells using a highly powerful microscope, we would see them as spherical structures. Within each cell, we can identify the nucleus, the compartment that houses our genetic material, as well as various organelles, including the mitochondria, often referred to as the cell's powerhouses due to their role in energy production. Every cell is also enclosed by a lipid membrane, represented here as the yellow envelope around the cell. When observed under a microscope, human cells display a wide variety of shapes. Some are nearly spherical, while others are highly elongated or specialized in form, reflecting the diverse functions they perform within the body. Given that the human body consists of billions of individual cells, it is essential that they communicate efficiently with one another to maintain proper function and coordination. Fortunately, our cells are remarkably efficient communicators. They are highly effective both in sending and receiving signals, which allows for seamless coordination across the vast number of cells in the body. As a result, cellular communication typically functions smoothly, enabling a continuous flow of information and intense activity within each individual cell. As we now understand, the human body is not composed solely of human cells. In fact, we host 10 times as many bacteria that also are an important part of our biology. We have them basically covering most of our surfaces and also our intestines. Bacterial cells are also highly active communicators. They produce a wide range of signaling molecules, including gases, peptides, and other bioactive compounds, which enable them to interact not only with each other, but also directly with our human cells. This brings us back to the fundamental fact that both bacterial and human cells are ultimately composed of atoms with highly specific structures. We now understand that the electrons orbiting the nucleus of each atom generate fields of motion, sometimes referred to as toroidal or torsic fields. This area falls within a subfield of physics that has been extensively studied for several decades. It is understood that torsion fields arise around particles that exhibit spin or orbital motion, such as electrons. Building on these insights, a scientist named Dr. Nesterov, founder of the Institute of Practical Physiopsychophysics, developed a unique device based on the principles of torsion fields generated by the human body, both as a whole and within specific tissues. This innovative equipment enables us to observe and measure how the human body responds to external influences by detecting changes in its torsion fields. These measurements help us assess whether a particular interaction leads to a favorable or unfavorable physiological response. A significant portion of the knowledge required to develop such equipment originates from fundamental research in the field of particle physics. As is well known, numerous accelerators around the world are already conducting advanced studies in this area. A great deal of the medical equipment used today also originates from fundamental knowledge in particle physics. Technologies such as X-rays, gamma rays, PET, and CT scans have all been developed based on our understanding of the behavior and properties of elementary particles. An especially interesting field that has emerged from this area of research is proton therapy, a medical technique in which protons are directed into the human body to target diseased tissue. This approach allows for precise treatment while also enabling observation of the body's physiological response. Based on the body's response, we can obtain valuable diagnostic insights or physiological feedback, and at the same time, this approach can be employed as a therapeutic modality. A number of proton therapy centers have already been established across Europe, including countries such as France, Germany, Italy, Russia, the United Kingdom, and others. The development of this device is also grounded in the principles of particle physics, particularly in the understanding of how changes occur within torsion fields. The device operates by emitting frequencies in the range of 1.8 to 8.2 Hz. It then evaluates the body's response to these frequencies and generates a spectral curve. This curve allows us to identify specific variations and assess whether a particular tissue, organ, or cell is functioning within expected physiological parameters. While this device operates on a similar principle of frequency interaction, it differs significantly from technologies such as ultrasound. For instance, ultrasound imaging and therapeutic devices typically operate at frequencies in the kilohertz range, often around or above 20 kilohertz. In contrast, this device functions at much lower frequencies. One of its most advantageous features is that it is entirely non-invasive, involves no radiation exposure, and delivers results quickly. It is therefore particularly well suited for gaining deeper insights into the physiological state of the body. As we will demonstrate later, the device also allows us to observe how the body responds to external inputs, such as a nutritional supplement or a specific food group, by detecting measurable changes in torsion fields. This provides valuable information on how such substances interact with the body's internal environment. As mentioned earlier, it has been well established that each organ and tissue possesses a distinct torsion field and specific spectral entropy characteristics. The data measured from the body are then compared to a reference database. In this process, a signal is transmitted into the biological system, and an output signal is received in return. During the measurement, two spectral curves are generated, typically represented in blue and red. The degree of overlap between these two curves indicates the condition of the tissue or organ being assessed. The greater the overlap, the closer the system is to its optimal or healthy state. Furthermore, by analyzing the differences between the two curves, as well as their positioning within the reference grid, we can determine whether the body is responding to an acute condition or if the findings are more indicative of a chronic process. For the food industry sector, one of the most relevant features of this system is a specific module within the software known as the test function. This function allows us to place a sample, such as a food item, dietary supplement, or any other substance of interest, into a designated testing compartment. The system then measures the body's energetic or informational response to this substance by analyzing the resulting output signal. In just a few minutes, we will demonstrate how this process works in practice and explore the types of insights this technology can offer. Primarily, we see great value in this approach when developing targeted food products. For example, formulations intended for individuals with celiac disease or lactose intolerance. It is equally useful in the development of specialized food supplements aimed at supporting specific physiological systems, such as the nervous or cardiovascular system. With this technology, we can easily evaluate the potential effects of a given substance on particular body systems and identify where the greatest benefit may be achieved. For now, thank you for your attention. A live demonstration of the system will follow shortly.
Software of this device. The device comes with such a tester where we can put in samples or pieces of food, and this biofeedback, they look like headphones, but they actually go to the temples. So we put these on. We put them on the head so that these plates are placed on the temples of the head. Once this is set, the machine firstly analyzes, runs through a scan of the body. And then once this is done, we can choose, for example, I have brought some samples here of food supplements, and we can just simply see what they do. For example, this is a propolis spray. We can put it as such if they are in inert packaging, such as plastic or glass. We can put it directly into this testing tube and click Vegeto test. It first asks for a name, so we'll say propolis and OK. And it will run through another scan where it will compare what kind of effect this substance has on the thoracic field. And this takes maybe a minute or so. And then it asks if you want to have a comparative analysis. We say yes. And here we can see that there is a strengthening reaction of 59%. Here we can also choose specifically, we can look at the arteries individually, or we can look at the entire cardiovascular system. We can also look at the nervous system independently, or the lymph system independently. And in this kind of way, we can see what kind of, we can see here on the images already, what's the difference between these segments. And then also here, it calls for like a percentage of it. So this is, for example, one example. We can test, for example, another. This is a solution of zinc that many times we are lacking, especially in winter. We repeat the process. We click the get to test. We say zinc. It runs through another scan. When we do these scans, we can also choose, this time the scan is running through entire body. We can also choose very specific areas or very specific sections, even very specific cells. For example, we could choose specific immune cells, or we could choose the nervous system as such. And again, we have strengthening reaction, and we can look at, for example, the muscles individually, or any kind of system, that organs, for example, internal organs, we see where zinc would be, would produce strengthening or improvement of the current condition. And in this way, we can just simply choose different food groups, food supplements, and compare their effects on the human body.